Okay, so welcome. So I am the host of today's colloquium, the first of the year. Also happen to be the chair of the colloquium committee. People aren't. No, it's on. Okay. I also happen to be the chair of the colloquium committee with Sid Goyal. So throughout the year, if you have nice things to say about the colloquium, don't hesitate to email me. If you would like things to change, if you have a blame, send them to Sid. Yeah, let me start with a land acknowledgement just to kick off this series. So I wish to acknowledge this land on which the University of Toronto operates. For thousands of years, it has been the traditional land of the Huron-Wendat, the Seneca, and the Mississaugas of the Credit. Today, this meeting place is still the home to many Indigenous people from across Turtle Island, and we are grateful to have the opportunity to work on this land. Okay, so without further ado, let me introduce today's speaker, the first of the year, Mara Freilich. So Mara has an undergraduate degree from applied math, and then she went to grad school at MIT and Woodsole to study physical oceanography. But after a few months, she realized she really wanted to learn biology. So then she went to Chile to learn biology for a year. Once she was telling me about a cruise, and she was telling me about how she was operating this instrument. I don't remember what it was to measure currents. And then she was like, but then I had to spend the day in the wet lab. She was actually like with a white coat and glasses and like manipulating biological stuff. So I asked her, do you know anyone who can do both, like physics and biology like that? She told me, I think there's someone in Oregon. So very unique profile. But then she went back to MIT to graduate. That was in 2021. Then she did a postdoc at the Scripps Institution of Oceanography at the UC San Diego, and started her job at Brown. As you can see, there's two departments, Applied Mathematics and Earth, Environmental, and Planetary Sciences. That was a year ago. She already has many projects going on. There's even more. She was telling me at lunch about working with communities and farmer communities in Southern California. So very broad person. So I'm very happy that she's kicking off this series, and please welcome Mara.
Yeah, so thank you for that very kind introduction, and thank you all for coming. I'm looking forward to hearing from you. Also, have any questions at the end and having conversations with many of you while I'm visiting. And so the title of this is Life in Motion from Turbulence to Climate. And what I'll be talking about is looking at this image in the background as inspiration and satellite. So this is a satellite image. The darker green color show areas where there's more chlorophyll in the water, which is areas where there's more algae, higher biological productivity. And looking at images like this was one of the things that really inspired me to study oceanography and the general subfield of oceanography, where there are many other people making important contributions, is biophysical interactions. And so thinking about how does the circulation of the ocean and the fact that the ocean is a moving fluid affect life in the ocean, where the vast majority of the primary productivity is carried out by single-celled organisms that can't swim faster than ocean currents. And so where they go is shaped by those currents. And one thing that's, I think, very clear from this image, and I'm going to be showing observations from this date and this region later, is that there are really multiple scales of motion, right? So on the 100-kilometer scale, we see that there's higher biological productivity near the coast and much weaker biological productivity offshore. But if we look at really the smallest scales, you can see swirls on scales below 1 kilometer, suggesting that physics at these scales are also shaping the fate, diversity, and distribution of biological productivity. And so I'm going to take a step back, explain biological productivity in the ocean, what drives it. And so here, this is, say, ocean water column, and the key facts are that light does not penetrate very far into the ocean. Depending on where you are, maybe the upper 100 meters of the water column have enough light for photosynthesis. Those upper 100 meters tend to have relatively low nutrients. That's the purple curve here, but relatively high carbon, relatively high chlorophyll, and relatively high oxygen. And so the physics plays a key role in this, but so does the biology. And so the reason why nutrients are so low in the upper ocean is because biological organisms using sunlight to perform photosynthesis are consuming those nutrients, turning them into carbon. But a key question that sets how much biological productivity we have in the upper ocean is how energetic physics are in supplying the nutrients to the upper ocean, right? So if there's relatively high nutrients at depth and relatively low nutrients at the surface, any vertical velocity or vertical flux, even if it just moves those nutrients a couple of meters, can result in substantially more biological productivity. And so that nutrient supply sets what we call new production. How much new nutrient supply is available to fuel this loop of photosynthesis and respiration in the upper ocean. The other question, and the one that I'm going to deal with a little bit more in this talk, is biological export. So how much of this carbon that's being fixed, so converted from inorganic carbon to particulate carbon in these phytoplankton cells, is able to go from the upper ocean to the deep ocean? And the paradigm had really been that this export production, so while there have been studies on this new production being driven by physics, that physics played an important role in supplying nutrients to the upper ocean, the paradigm on this export production branch had really been that it was driven just by sinking, so settling out of cells from the sea surface, without as much consideration for the role of physics in that process. And so just to not mislead too much about carbon in the ocean, there are multiple pools of carbon as we think of them, but I'm really just going to divide them into an organic pool, so carbon that has been modified in some way by biological organisms, and what we call a dissolved pool, which is just carbon dioxide that's been dissolved into seawater. There's vastly more carbon in that dissolved pool, the carbon dioxide that's been dissolved in seawater, than there is in this organic pool. I'm not going to be talking about this dissolved pool at all, though. I'll just be talking about what's been modified by biological organisms. And there has been some work on the role of physical processes actually in both of these processes. How do you bring inorganic carbon to depth, but also how do you bring organic carbon to depth? And so what I'm going to be talking about today is what is the role of ocean hotspots? So what's the role of these small scale swirls we see in biogeochemical fluxes? And by that, I mean fluxes of biology, but also geochemistry, so things like carbon and nutrients, and that really there are interactions between them. And so the plan today, I'm going to very briefly touch on biology at a fairly high level. I know this is a physics audience, but it really as motivation for going back out into the field and studying physics in detail. So first, I'll talk about fronts and biological fluxes, and I'll define these terms. Then I will talk about really, what are the physics? What are the physical oceanographic dynamics at fronts? And then bring us back into big picture again. Think about what are the impacts of climate change on these sorts of fluxes. Okay, so what is a front? A front is a meeting of two different water masses, one that's less dense and one that's more dense. And there is substantial potential energy in this configuration where the denser water will slide below the less dense water. Because of the constraints of rotation, the Earth is rotating, this will not happen in a smooth way. Instead, there will be eddies and significant motion along what we call density surfaces. So this interface between the less dense and the more dense water, I'll refer to as an isopycnal or constant density surface. And if we had, say, a biological organism, this green parcel in the surface, we're asking how is it being moved downwards, say out of the sunlight. And so that's a simple view. Now, if we layer biology on top of that physical picture and add a ship to signify we were going out to sea to sample these sorts of things, what we see is what I was showing in that 1D schematic, right? That there's relatively high green or biological productivity in the upper ocean. But in the schematic view, I'm showing the results of our study, which is that we found these streamers of high chlorophyll, high carbon water being carried into the ocean interior. And so just to give evidence that this is not just a schematic, again, green is chlorophyll, and here the black lines are density surfaces. So the density surfaces are plunging downwards. This is over a distance of just tens of kilometers, which for oceanography is fairly small distances. And in the upper 200 meters, and we see these plumes of high chlorophyll. So this is just one example of the many. You could think of this, say, as just a 2D face of this schematic diagram. And so I went to sea and collected, in this case, 224 samples that we analyzed for community composition. One of the important things as I show you the results going forward is we actually did 3 different field campaigns. So we were 3 different seasons. And so this process really repeated itself many times. This process of transporting the carbon from the surface into the interior. And so this is what our sampling plan looked like, right? So 3 different field campaigns spanning from March all the way through July. So from springtime into the summer. And before I go too much farther into the results and what we found while we were at sea, I want to acknowledge the many people that it takes to make something like this happen, right? So whether it's a sunny day where the entire science party is what we call ourselves, the entire science staff of the ship gets together to take a photo, or sampling late at night in bad weather and waves, it takes an enormous number of people. So this is not just my work that I'm showing. And I'm going to show when I said I sampled community composition, I filtered water on board the ship, and then analyzed the DNA back in the lab to look at what the community composition is. Right? And so you don't need to know the details of it, but just to emphasize how I know the diversity of organisms in the water. And so to give a broad view of what we found, so the typical conditions, I mean, we found fairly typical conditions. So in these surface waters, and I'm just showing these on a schematic, in the surface waters, we found phytoplankton, which is what we expected, right? There's enough light to perform photosynthesis. And so phytoplankton, which are these algae or planktonic organisms that can perform photosynthesis, or abundant in that upper layer. In the deeper layer, we did not find, in a, say, typical scenario, you would not expect to find phytoplankton. You would not expect to find organisms that are primarily performing photosynthesis in the layers where there's not enough sunlight for growth. And so what was really interesting to me then was the bacteria. Not a lot is known about many marine bacteria, and so don't have to worry too much about the details of these marine bacteria, but typically the bacteria actually has a depth structure as well. And so in the upper ocean, we found the communities were dominated by this bacteria, SAR11-1A. Well, in the deeper ocean, there was this distinct bacterial community dominated by a different clade of SAR11, and by other clade, SAR324 was the other dominant organism. When we identified these filaments, which we were able to do using underway observations on board the ship, so identified these high chlorophyll filaments, sampled the community composition in them, had enough that we could perform statistics what we found was that these communities were dominated by phytoplankton and SAR 11.1a, and in a statistical sense, they were much more similar to surface communities than other communities at the same depth. But there were some changes, and I think those are fascinating as well. And in particular, we found changes in the bacterial composition where there were some bacteria that seemed to thrive on being transported into the dark. Not a lot is known about them. Here I've highlighted the top, or the two most abundant, SAR 406 and OCS 118. Not a lot is known about these bacteria, right? They've been found in other environments, but in places they've been found, it was after decaying phytoplankton blooms. And so this is suggesting maybe the plankton, although we found them there, maybe they're not doing very well because they're in the dark, and instead these bacteria are taking over. And the significance of this is we're not only seeing carbon being carried downwards, but we're seeing it carried downwards as part of a whole community where it's being actively transformed. And so how significant is this process? How significant, so I've said, we see these streamers of high chlorophyll going to depth. Does it matter? So remember in my introduction, I said the paradigm before had been that sinking, right? So just cells settling out, right? They're somewhat negatively buoyant. They settle out at rates of 1 meter per day to maybe 100 meters per day. How much carbon does that bring to depth? So that's these orange bars here. They don't have error bars associated with them in the previously published estimates. Here's a map where I computed using existing estimates for the most part, with some modifications, how much carbon could be transported by frontal dynamics, right? So I took parameterizations that are used in models for the impact of fronts on tracer fluxes, and I said, well, let's apply that to global observations of fronts, right? So how much these density surfaces are tilting, and global observations of carbon, and came up with this map of fluxes here. And you can compare it in those green bars. How much is this frontal subduction contributing to carbon export? So the error bars are pretty large, we acknowledge first of all. And that's largely because we have very few observations of subsurface carbon. These sinking estimates, they rely on surface carbon. For these estimates that I'm making, we really need subsurface carbon observations, right? So we need more observations. Oceanographers will often say that, right? But even accounting for these large error bars, we can see that these fluxes are quite significant, even compared to sinking fluxes. So we think about half the carbon that's being exported or that's being transported into the interior could be because of these physical processes rather than solely biological processes, is sinking. But how is it getting down? Right? So this motivates us to understand the physics of fronts better, and in particular, the physics of vertical velocity in the ocean. So I want you to note, so this is a model run. So I initialize the model based on the observations and then run it forward. Vertical velocity is extremely challenging to measure in the ocean, right? So the model vertical velocity, this is right? So the largest vertical velocities I have here are about 20 meters per day. The largest observed vertical velocities are about 1,000 meters per day. More typical large vertical velocities would be closer to order of 100 meters per day, right? So vertical velocity is very slow, right? If you were to walk just 20 meters per day, it's extremely difficult to measure that because of the noise of the sensors we have, right? So this is largely within the noise limit. But in models, we are able to see vertical velocity. And so you see areas of upwelling and downwelling. Here, the black lines, again, are density surfaces, right? So upwelling and downwelling associated with these density surfaces, some resulting in downwelling, so downwelling of dense water, upwelling of less dense water. And so to see how water is getting from the surface or near surface into the interior, I used what we call particles, so I initialized particles in this model. And here we're looking just at one density surface, so we can see how those particles are moving downwards along those density surfaces. And the key thing for where we're going with this talk is that the vast majority of the water parcels, so here I'm showing snapshots from just 3 different days. Now we're looking down, say on the top of that model. The lines again are showing density surfaces, but these dots are colored to show where water parcels were when they left the surface. So nearly all the water parcels that leave the surface are at these fronts, right? Clustered at these fronts. They all have positive relative vorticity. And so this is, say, the magnitude of nonlinear terms in the equations of fluid motion. But they're not leaving the surface everywhere along these fronts. They're leaving in certain patches. And so this is something that motivates us to understand the dynamics of these fronts in much better detail, right? So this is a model with 100-kilometer grid spacing. We say water parcels. So that global estimate I showed you, I use data at about 1 degree. So that's about 100 kilometers, right? So it's fairly coarse and doesn't account for this fact that here my model domain is about 125 kilometers, right? So water is not leaving everywhere within this model domain. Instead, it's really at these areas of the tightest density gradients where it's leaving the surface. But so the key takeaway from this, say, motivation section is that subduction, so this process of water leaving the surface and export of whole microbial communities by a process that includes the mesoscale, so scales of about 100 kilometers, and submesoscale scales of about 1 kilometer, contributes to carbon export. The fluxes occur in a disproportionately small area, and there was a significance to looking at the community composition, that there was active biological, both planktonic and bacterial transformations occurring during this subduction. Right, but I mentioned, so now we really want to look at like, what are the dynamics of these fronts? What are happening on these scales of just a couple of kilometers where water parcels are leaving the sea surface? Right, and so here, I won't get into this in detail, but just to put up the equations of fluid motion here, pointing out that I'm talking about rotating fluid dynamics. But instead, I use that word submesoscale. So I'm going to introduce this concept of scales. And I'm going to show a few plots that are in wave number. And so in those, the largest scales are going to be on the left-hand side with the smallest scales on the right-hand side. And these scales of about 1 kilometer are what we call submesoscale, and scales of like 10 to 100 kilometers are what we call a mesoscale. And the mesoscale is in a balance between the pressure gradient and rotation, the inertial terms. And so at the relatively large scale, so the key question now is what's happening at fronts? And one way that we're going to approach that is to look at transfer of energy. So now we have transitioned to entirely physics. We're going to leave biology behind for a moment. And so the question here is, so if we have injection of energy at large scales, it goes to smaller scales to create eddies. But then there's a transition back to larger scales where the energy that's put in, say by wind forcing or tides, could be dissipated at the bottom of the ocean. The other question, right, so it could be dissipated based on bottom friction, but there's a surface dissipation. How do we get energy to surface dissipation? So to do that, there's another part of this where we bring energy in what we call a forward cascade from these largest scales that are the scales of mesoscale eddies down to smaller spatial scales. At these scales, there's also a transfer of energy back to larger scales in what we call an inverse cascade. But if we have surface dissipation, so this is by 3-dimensional turbulence of the sea surface, there must be a route to bring energy from the largest spatial scales all the way to the smallest spatial scales without transferring it back upscale. Right, so the other sort of introduction about these scales is, so I typically call wave number K, and there's power laws of distribution of kinetic energy across scales. Where at these largest scales, we have approximately K to the -3 spectrum. I'll remind you about these as they come up, at K to the -2 at these intermediate scales. And then we think three-dimensional turbulence without constraints of rotation has about K to the -5/3. They're very close to K to the -2. Right, but so this, now we're turning to this idea of a forward energy cascade, right? So have we observed this cascade of energy from larger spatial scales to smaller spatial scales? So what I'm showing here is that energy flux as a function of wave number here. And these are some relatively recent results. The first one on the left from observations, showing that there is a positive flux, which here is forward, so from large scales to small scales, at the smallest spatial scales that have been observed. So this is at scales of less than 1 kilometer. This has been shown in models as well. So these are actually both from the Gulf of Mexico. The models are only barely resolving this forward flux. And so the way that they and I compute these energy cascades is by filtering these equations of motion that I showed you before. And so doing a filter at a particular spatial scale, right? So this is sort of average, is rigorously derived averaging at a particular spatial scale where anything below that scale is not considered. Right? And so if we're looking at flux across 1 kilometer, we do this averaging at a 1 kilometer scale. And so I'm going to do this in observations as well, look at the energy fluxes. And one thing, so the previous the previous observational study, they were able to establish that there is a forward energy flux, but not where it's happening or what dynamics are driving it. And to do that, I think we needed new types of observations that we haven't had previously. And so I was fortunate to be part of the submesoscale ocean dynamics experiment, which is a NASA-funded field campaign that took observations in 2021 through 2023, using a whole host of platforms with a large number of PIs contributing. I'm going to focus actually on observations from airplanes, in particular from the smallest airplane involved, which is called the SIO MASS, and an instrument called Doppler scat, which is a Doppler scatterometer. Both are able to give us direct velocity observations, really for the first time at the 100-meter scales. And so here's a region. We were operating off the coast of California. Here we're looking at satellite sea surface temperature. With the planes, the planes fly in these ground tracks where they measure a number of properties in addition to direct observations of surface velocity. And if we zoom in just on one front within this whole observational set, here I'm showing relative vorticity. So we're able to compute, we not only have observations on these tracks, but there's some cross swath width, so we can compute velocity gradient quantities like vertical velocity, but also here I'm showing the relative vorticity. And so the first thing we look at is what is the distribution of energy across scales? And so this is a kinetic energy spectrum, right? So here again is wave number K. So from 100-kilometer scale to 1-kilometer scale, the amount of kinetic energy contained in each scale. And this has a K to the -2 spectrum, more or less, right? We have error bars that I think are somewhat difficult to see here, but you can't really distinguish K to the -5/3 and K to the -2 in these sorts of observations. But what's significant about these observations, and in particular the observations in the solid line compared to those from the dashed line, is that we're able to push the spatial scales that we can see to much smaller spatial scales that we were able to observe previously from ships. And we're able to do it much faster. But this k to the -2 slope doesn't tell us a lot about the dynamics, right? It could be because of internal wave motions. It could be because of fronts. And it could be because of a dynamics called surface quasi-geostrophy that has to do with surface fronts, but is associated instead with a k to the -5/3 spectrum. Another thing I want to point out is that these observations are pushing us below the scale of the state-of-the-art global models, right? So the state-of-the-art global model has about a 2-kilometer grid spacing, but is not able to resolve dynamics much smaller than 10 kilometers. And so we were inspired by these models to go out and take these observations, but now we're able to push to smaller scales than the models. I think we also, as we're observing these smaller scales, now need new methods to understand the dynamics at these scales, right? So we've traditionally looked at those kinetic energy spectra. There have been a substantial interest in separating waves from what we call vortical motions or quasi-balanced motions. But one of the arguments of those sorts of models is that the flow is fairly linear, where you have linear waves that are just superimposed on vortices. Part of the problem as we get to these smaller spatial scales is that the velocity derivatives becomes much larger, the flow becomes much more nonlinear, and we have much more vertical velocity. And so these traditional methods weren't working. And so there's a method where people typically apply to these sorts of observations where they are able to separate waves and vortices. I derived a new method that allows us instead to turn this on its head and say, well, when this method fails, right? So where this assumption that the stream function, which is the balanced part of the flow, and the potential, which is the unbalanced or the part of the flow that's associated with vertical velocity, become correlated, as they do at fronts, then the flow is relatively nonlinear. So if these two are uncorrelated, if the stream function and potential are uncorrelated, then the cross spectrum between a long track and a cross track velocity here is real. If not, then they become complex, right? So I'm going to test for flow transitioning to nonlinear frontal dynamics based on where this cross spectrum becomes complex. The assumption here is that there is significant coherence between the long track and the cross track components of velocity, right? So I'll show that first. So that across our observations, and here I'm showing two different field campaigns, one that was in more eddy-like area and one that was in more frontal-like area, but both actually relatively weak flow compared to the global ocean. And here the coherence of the long track and a cross track velocity, so you can think of this as north-south velocity, is significant. And so then I look at the cross spectrum, and this is what's really exciting, which is at the cross spectrum phase, we see that at largest scales, this cross spectrum was real, and at our smaller spatial scales, it transitions to being imaginary, which suggests that we have that the long track, or that the velocity potential and stream function now become correlated with each other at the smallest spatial scales. And this is what we would expect if there's significant vertical velocity associated not with waves, but with, say, straining motion of the frontal flow itself. Right, so we derived a new method where we're able to detect this transition in the dynamics at these smallest spatial scales, so below 10 kilometers or below 6 kilometers, so scales smaller than 6 kilometers in their frontal area. And so we're able to rule out internal waves as the main driving mechanism of our spectral slope. We can also, so I ran a model of surface quasi-geostrophy, and this is just showing one example where we do not see this transition in surface quasi-geostrophic models that we see in the observations, right? So this is saying that the surface quasi-geostrophy is also probably not the leading mechanism for the distribution of kinetic energy at the smallest spatial scales in our observations either. And so the suggestion from this analysis is that ageostrophic frontal dynamics, and I think I set you all up from this by saying I'm very interested in fronts, right? That these frontal dynamics that are not associated with balance flow are the dominant dynamics at the observed scales, right? So these newly observed scales below 10 kilometers or smaller than 10 kilometers. The other really exciting thing that we're able to do with these observations, and here I'm just showing a small snapshot, but is to show where we have kinetic energy fluxes. So here below, I'm showing our swath, right? So here I'm just showing a 4-kilometer patch of the ocean, although we did this analysis over much larger scales. So in our 4-kilometer patch, I computed the kinetic energy flux. So we have some error in our observations, so I'm trying to be fairly conservative. This is a triple correlation, so we have fairly large error bars, but this blue curve here is showing that the kinetic energy flux becomes significantly positive right at the fronts. And this was true across the observations, that right at the fronts, that's where we have kinetic energy going to smaller spatial scales, where we're not observing the dissipation directly here, but where it could potentially be dissipated by surface turbulence. And so this is giving us true lines of evidence that frontal dynamics are really driving the distribution of currents at the smallest spatial scales in the ocean. And so from this section, I showed that we have a new use of the cross spectrum to determine dynamics. And across these observations, not just in the detailed section I showed here, but across the multiple observations we took, really a predominance of ageostrophic frontal dynamics in these observations, which again are off the coast of California. And showing that not only does there exist a forward kinetic energy flux in the surface ocean, but that it's localized at fronts. And I didn't show this in detail, but we were able to go a step farther and show that it's associated with shear instabilities and velocity covariance. Okay, but how to, right, so that's pretty detailed, right? Like studying the smallest fronts in the ocean, saying what are the dynamics there, right? How does this matter? So the first way that I'm going to talk about how this matters is again for biology, returning to our original motivation. And so this is one of those fronts. It's a front that's fairly similar to the one I showed previously. So here's the density step, right? So we're back here, we had this gradient in temperature. Here I'm showing a gradient in density. And here we have high chlorophyll right within that frontal zone, right? So these fronts are clearly associated with biological processes as well in the observations. Returning to our motivating image from the beginning, I'm going to actually zoom in on an eddy. So this really high chlorophyll patch here. On the ship, we spent a little more than a week sampling that particular eddy. And this is what that eddy looks like from the planes, right? So this is a satellite. This is a plane. For oceanographer, just seeing these images that the planes were able to provide us was incredible, right? This looks like a model output. It's not. These are observations. And so this is relative vorticity. So here you can think of that as the spin associated with the velocities. And so this eddy is spinning in a cyclonic or counterclockwise fashion. So it has a positive relative vorticity associated with it. There's a whole bunch of other features, right? There's many other things going on here on fairly small spatial scales. For the planes, we're able to sample this eddy day after day after day, right? So these are 5 snapshots of that same eddy. I'm showing these 5 snapshots because you can see it's deforming a lot over the course of a couple of days, right? So it's not, say, a totally balanced or steady type feature. What I did working with graduate student Elise Boudin at Brown is just composite those 5 days. And then we get a pretty circular looking eddy, not perfect in terms of the relative vorticity here. And this is really small, right? So this is, here the box is 12 kilometers. This is an eddy is about 8 kilometers. So truly within our submesoscale or less than, smaller than 10 kilometer scale. Well, I said previously that vertical velocity and these energy fluxes are associated with fronts, which are areas of sloping density gradients. These observations now are beginning to challenge that. And they're not inconsistent with the models, but here I'm plotting the divergence. So again, remarkable, surface divergence is associated with surface vertical velocity. So remarkable in that now we're able to directly observe vertical velocity instead of having to model it. But we see that while the eddy looked pretty circular in vorticity, this divergence was kind of everywhere, or it was not associated. We have some downwelling right at the beginning, but at this northern edge that was interacting with other features appeared to have the largest vertical velocity associated with it, right? So this idea, vertical velocity, these sorts of fluxes, whether they're baroclinics associated with density gradients or barotropic, are really localized. The carbon flux, so we were measuring carbon at the same time, we have that chlorophyll image. The carbon flux associated with this feature would have been about 300 milligrams per meter squared per day. And this is really significant, right? So we would have, those sinking fluxes that I showed previously, those would have been about 100 milligram per meter squared per day, right? So even in this average sense, significant carbon export coming from this feature. Here's what it looks like in the chlorophyll. So again, you see it deforming. We took a bunch of samples on this feature. And we can combine both the plane-based observation and our sampling to look at the rate of change of chlorophyll from at the sea surface. And so I said this whole thing, there's a downward vertical velocity associated with it. So we're losing some carbon from that process. These values, so the purple here is negative, so we're losing carbon, green is positive, so the community is growing. What we find is that on the whole, this community is decaying, probably actually majority from biological processes with a small contribution from physics, but right on the edge of this feature, right? So right where we think frontal dynamics would matter most, we see some growth. So this is suggesting there's perhaps upward supply of nutrients right on that edge. And so here, just to make the point that these submesoscale dynamics shape the fate of carbon and the distribution of microbial communities. And I would be happy to talk more about the biophysical interactions that we saw in this feature. But one of our conclusions here was that although the physics are significant, the biological dynamics are actually dissipating carbon much more rapidly at the sea surface. But the rate and spatial distribution of that dissipation was shaped by submesoscale dynamics. And that submesoscale dynamics, so this feature was moving westward very rapidly, so offshore. So submesoscale dynamics are really important for that offshore transport, so lateral redistribution in addition to their vertical flux. Right, so then thinking about climate change, there's a number of impacts that we think about when we think about climate change, right? So it's a somewhat abstracted way to think about it, to just think about ocean's role in the carbon cycle. In a concrete way, right, we associate climate change with fire, storms, floods, heat waves, and each of those really does have an impact on the ocean as well. And so while we were out sampling in April 2023, this was an extreme water year in California. Where a number of communities were really deeply impacted by flooding that they experienced from a succession of atmospheric rivers that hit the coast. And we expect, while California overall is in a drought, we expect these extremely wet years to increase in frequency in California. And so climate change is associated with more precipitation extremes, in addition to a change in average precipitation. And so an increase in extremely wet seasons, and in that same model ensemble, I was able to show that that increase in extremely wet seasons will also result in extreme springtime discharge as well, right? So these are projections of proportion of frequency of extremely wet seasons and proportion of high discharge seasons. So the eddy I was talking to you about and these observations from 2023 then were impacted by this discharge. We saw, while we expected high chlorophyll water actually to be associated with relatively salty water, that came from upwelling of deep waters. Instead, once we got out into the field, we realized this high chlorophyll water in large part was associated with relatively fresh water that we think came from rivers in the coastal area. And so this year, 2023, was also extreme in that it was relatively fresh compared to the historic satellite observations. And when I say high chlorophyll from the satellite, this was so high that you could see it visually. So this is a photo that one of our collaborators just took out of the plane with his iPhone camera, right? So you can see the change in color of the surface that satellites are able to detect, but we could see it visually that this was fairly scummy looking water out in the open ocean. And these observations were also anomalous in other ways, right? So this is salinity, and this is the ratio of nitrate to phosphate. Biologically, so due to biological transformation, we expect that ratio to be about 16. The gray shows all historic observations since 1957 in the same region. And in the green, those are our observations, right? So our observations are much fresher, as you can see. But in this particular eddy that I was telling you the story of, where we have quite a few samples, they're also extremely high, but it also have a change in this nutrient ratio with much more phosphate than nitrate. So like human wastewater is associated with excess phosphate. And so based on Lagrangian backtracking, I think this feature came from the San Francisco Bay. And so these biogeochemical observations are consistent with that. Right, so what we were able to study, like how do submesoscale dynamics work in this particular freshwater feature? In a larger context, it's telling us something about how the ocean might respond to climate change and how the way that how water policy in California may influence not just California, but also the open ocean as well. Lastly, and I'll just touch on this briefly, because I think it's some exciting new work that I'm doing with postdoc Lily Dove in my group, is also thinking about as the ocean changes in a global sense, how might these carbon export processes change? If we think that carbon export is significantly driven not just by sinking, but also by physical processes, how might that change as the ocean warms? So as the oceans warm and actually as winds increase, observations are showing that in some regions, particularly the Southern Ocean, the stratification is increasing, right? So that's the intensity of the density layering, right? And as that stratification increases, we expect that it might be harder to transport water from the surface into the interior, potentially reducing carbon fluxes. So we did a modeling study together where here we varied stratification using the same sort of idealized model that I showed you previously and look at the tracer fluxes. So these tracer fluxes are negative, meaning that they're downward into the ocean. So a larger negative flux means more carbon export. And so here, this is the sort of current stratification conditions for the Southern Ocean. If we decrease stratification, we would have more downward flux. If we increase stratification, we can expect for some time to decrease that flux. But our results are actually suggesting that there might be a plateauing effect where we'll decrease stratification if we decrease flux if we increase the stratification slightly, but not if we continue to increase stratification beyond that. So there are two lines. I was looking primarily at the solid lines when I said that, and these solid lines are flux at a variety of different depths in the ocean. These dashed lines are also if you change the slope of the density surfaces, and if you allow those density surfaces to change slope as well, we find that we're actually relatively insensitive to stratification if we're near the current conditions, right? So just saying that we need improved understanding here. One of the things I want to highlight about these results is we're finding that this has to do with fronts in the upper ocean. But once we're deeper, it actually has to do with coherent vortices, much more similar to the eddy I was showing you at the sea surface, and much less with fronts. And this was one thing that was really surprising to us that we were able to pull out from these observations. And that's the reason for the difference between these two scenarios is the extent to which the coherent vortices matter. So what we found, first of all, emphasizing from an observational aspect that these collocated and coincident observations of velocity and tracer distributions like carbon, but also rates of change from biology are really as essential to painting a full picture and truly quantifying the impacts of eddies on the ocean carbon cycle and the role of oceans in climate. I showed from a physical perspective a predominance of ageostrophic or unbalanced frontal dynamics in observed submesoscale features, that the forward energy flux that has been found in models and other observations is really localized at fronts and associated with velocity covariance. And then again, in a bigger picture, why does this matter? These submesoscale dynamics are shaping the fate of carbon and distribution of microbial communities in the ocean. And they're mediating the response of ocean ecosystems to climate change as well. And so with that, thank you, and I'm happy to take any questions.